Thank you all very much. Thank you for listening, and thank you very much to the previous speakers. I want to ask a funny question on the border between very, very wet biology and very, very physics-based. I want to ask, why do cancer cells metastasize? What is it that pusheth them underneath? So I can let me just start off by thanking people. Andy, Moynihan and Martin up here did the melanoma work. Luke below did May's work in pancreatic cancer work and is a mathematician. John, Adam and Michele are mathematicians and an engineer respectively. And we will see their stuff later in this talk. Okay, and then a whole load of extra people helped. In particular, very sadly, Mike Wakelam, who died in the first week of lockdown of COVID, and we miss him no end. So my story starts with random migration. And when you look down a microscope at cells, especially if you cheat and use time-lapse, random migration looks very, very active. But actually, I think the sort of random migration we tend to see is very unlikely to make anyone metastasize. Why do I think that? The distance that cells move by random migration scales pretty accurately with the square of time. What that means is that what you see looking like rapid and efficient migration down a microscope over a short time does not result in any substantial migration over a long time. 10 times the distance, which is still not much in when you move from a microscope to a body, takes 100 times the time, and so on and so forth. The other thing is that cells are really good at finding an easy way. So unsteered migration does not take you through basement membranes, does not take you through endothelia. It certainly does not take you through the blood-brain barrier. So when you see cancer cells doing those things, you tend to conclude someone has steered these cancer cells to do that thing. So if you want to understand why cells do metastasis, you really need to understand what is steering them and why. And if you look at, for example, melanoma, quite a lot of the things I'm going to talk about are with melanoma, although this is very generally true. You can see that a melanoma is not spreading in a random fashion. You can see in particular at this edge here, random migration should give you a sort of Gaussian shape in 3 dimensions. Lots of the random migration takes you nowhere. The most extreme cells might come out to the edges. But these cells in this melanoma are not being steered. And these ones down here are coming out in a sort of ray pattern, which we, from a mathematical viewpoint, might expect, but is not predicted by random migration. If you look vertically, which is what really counts for the spread of the cancer too, also, you don't see a lump, you see a channel, you see a decision to spread vertically down into the patient, which is very, very bad indeed for a patient. And of course, those of you who've known me for a while will know that the answer is going to be chemotaxis. So here you see a chemotaxis chamber. On the left here, you see an area that's got low serum in it. On the right here, you see something that's got high serum in it. And you see that melanoma cells, pretty much every melanoma cell we've looked at, is really chemotactic. They really like to go to areas where there's high serum. And in the interests of time, I'll just tell you the key ingredient in these experiments turns out to be LPA, lysophosphatidic acid, which is a sort of demon mitogen and chemoattractant that's present at stupidly high levels in serum. Okay, so the question we're going to ask, again, we're asking about information here. We're not interested in pathways right now. We're asking, why are there gradients of chemotaxis? Why does chemotaxis matter to metastasis? Where does the gradient come from that is making a melanoma spread? You can ask it a different way if you want, which makes it clearer. Why does the body have an interest in establishing gradients in order to get cells out of a melanoma and into your brain? It doesn't make sense. And the answer to this came in a control experiment about 10 years ago now, which completely redirected the way my lab has worked ever since. And that goes like this. I showed you the movie before where you had low serum on this side and high serum on this side. What happens if you put high serum on both sides? Again, it's not symmetrical because the cells are only in this side, but what you see is the cells still spread just as much. The gradient of serum that we gave them was not important. What's important is that the cells were in the left and not in the right. There is still chemotaxis going on, and it's still chemotaxis to LPA, but the chemotaxis is not to a gradient that we have given the cells. It's chemotaxis to a gradient that the cells are creating themselves. And just to emphasize, you can even see this leading wave here has all of the gradient in it. All these cells behind the leading wave, there's no LPA left at all. There's no gradient. The information is only in that narrow band at the front, which is amazing. It's not the way we saw chemotaxis at all. So if you go and look at a bunch of cultured melanoma cells, you see down here in the red, they're not generating any LPA. You have to put that in the serum. But you have a dose-dependent ability of the cells to break down the LPA. A low confluence plate breaks down the LPA slowly, as shown in blue, and a high confluence plate will break down all of the LPA in a culture dish over about 24 hours. This is actually pretty important if you are culturing cells, because LPA is a really strong agent. It changes the shape of cells. It is a strong mitogen, and it changes the migration of cells. And if you are feeding your cells with fresh 10% serum, for about a day, they have a high level of LPA. And then by the end of about a day, they've broken it all down and it's not there anymore. We found the enzyme that is responsible for breaking down the LPA. It's LPP3. It's a well-known enzyme. It's on the surface of the melanoma cells on the outside facing out. So it's not breaking down anything on the inside of the cells. It's breaking down extracellular LPA signals to inactivate them. And you see here, this is a 2.5D assay. This is a circular invasion assay where the cells actually have to go punch their way through a matrix. And you see wild-type cells do it very, very efficiently. If you take out the cell's ability to respond to LPA, then they don't invade at all. They push forward a little bit as they grow, but they don't invade. And exactly the opposite of what you would have predicted beforehand happens. But what we now understand is there, which is if you take out the enzyme that breaks down the LPA, then the cells no longer invade. They no longer invade because they're not able to set up a gradient. So there's no chemotaxis because there's no information. So as well as LPA being essential for invasion, breaking down LPA is also essential for invasion because it's the gradient here across this cell monolayer that's important. It's not the molecule itself. The signal is the gradient in the molecule itself, which needs both a source and an enzyme to break it down. And this is cool. This really suits the sort of way we are thinking. There is a subset of referees absolutely hate it. They say either the LPA is there or it's not. If the cells are breaking it down, it's not there. And of course, this is totally missing the point. A gradient is an evanescent thing. It's made by flow. So you need to break down LPA continually in order to respond to it. This is true in 3D as well. Here is a melanoma organoid. It's not really an organoid because it's just a lump of melanoma cells. But if you put that in matrix that's got serum in it, it spreads out like mad. But if you stop it from breaking down the LPA, which is the chemoattractant, it doesn't spread out as much anymore. There's no longer any gradient there, so the cells mostly just sit pat. And finally, the most remarkable demonstration here is that other labs were doing a whole load of experiments on a melanoma model of mice. And we took the tumors from the mice when they killed them before they suffered. And we did a core through the middle of each tumor, and we did a core on the edge of each tumor, and we did a core just outside it. And lo and behold, in every tumor we examined, there is a gradient of LPA. There is a gradient that's high on the outside, that's low on the inside. So the gradient is pulling those melanoma cells out from the tumor into the rest of the body. So we've shown that pretty much all melanoma cells we've looked at are highly chemotactic. We've shown that they generate their own gradients, and we've shown that those gradients exist in vivo. So actually, this story where the cells make their own gradients is really, really, really important in what is making the cells metastasize. So the model goes something like this for why you need to catch a tumor early. If you have a small tumor, it breaks down a small amount of the local attractants, LPA, for example. Because it only breaks down a small amount, there is no gradient and the tumor doesn't spread. But there is some magic size, and when a tumor reaches that magic size, then the cells at the edge of the tumor experience a gradient. And when the cells experience a chemotactic gradient, there is a drive, there is information trying to make them migrate out of the tumor and into the rest of the animal. And that is one of the key drivers of metastasis. It also explains a number of things. It explains why in melanoma, the key parameter is how thick the melanoma is. Unlike, for example, breast tumors, where the stage of differentiation of the cells is what's key, in melanoma, really the key thing is this vertical thickness. And that vertical thickness is telling you whether or not the cell is able to establish a gradient sending the cells into the body. Also explains one or two other funny things. It was long observed this thing called Prejudice Spread, where melanoma cells spread upwards into the epidermis. And that had never really made sense. If you have something that's being encouraged by the body to move from the tumor into the body by a gradient, then why would it spread upwards? But if the information comes from the melanoma cells, the tumor, and makes it spread outwards, then of course they'll go up into the epidermis as well as down into the bloodstream and so forth. Okay, so what this makes us all conclude is that the idea of self-generated gradients, that actually in chemotaxis, some of the most important gradients are the ones that are made by the cells themselves. Self-generated chemotaxis is terribly hard to measure. It's very, very local. I showed you in my chamber assays, the gradient we think only exists over about 50 microns, something like that, 100 microns. It's driven by diffusion. So if you stop for a moment to try and study it, then new chemoattractant has diffused in and your gradient no longer looks the same. It's very, very short-lived. If you remove the cells, it's gone. The big thing that everyone tells me I should be doing is looking at fluorescence ligands. But of course, if you take fluorescent LPA that has got a fluorescence tag on its lipid tail, and LPP3 acts on it by chopping the phosphate off, it's still just a fluorescent. It doesn't look any different at all. And there is not, at the moment, a ligand that is both fluorescent and also whose fluorescence changes when the LPA is inactivated, but which also activates the receptor. So it's a really tough thing to do. Although we are still trying, we are using G protein reporters. They're really difficult to use, but we think they are going to work. But the best way of studying these things in the meantime is to use detailed computational models of different kinds. And when you have detailed computational models, you look and see if they're right. We have quite a lot of measurements and quite a lot of parameters. And in general, we do a pretty good job. So here's an agent-based model. The red is the attractant. The cells are starting there at the left. Now, agent-based models are lovely. They let us do complicated things with complicated rules. They're not very rigorous. You can do things that are not really justified by physics. We use them occasionally, but I'm going to use tougher models later. But this shows absolutely beautifully what we think is going on here. The cells follow a gradient. As the cells migrate, the gradient migrates. the concentration is way saturating to start with, lots of attractant. It doesn't need to start in a gradient, to reiterate. The gradient comes from the cells, and the cells are responding to it, so it's a positive feedback mechanism of quite delicious complexity, and the information is all coming from the cells and the cells only in this case. Now, I haven't got time to talk you through this. I've used much of my time already, but I'd like just to ask, why would cells want to use these things? Well, there's lots of reasons, but here are two of them. Firstly, if you imagine cells chemotaxing over 1 millimeter, the top here, we're going to show cells in an imposed gradient. So there's a source of gradient here, and these cells are just going to respond to it. And what happens if the cells start responding? And then as the concentration gradient gets higher, they get less and less information as they get further along in the gradient, I should say, because the difference in receptor occupancy across each cell gets less and less. And actually, it turns out that you can almost never chemotax across a 1 millimeter gradient. You either have too little for the cells to detect at the low end, or no information at the high end because the receptors are starting to get full and there's no difference. In a self-generated gradient, and this now is a PDE-based model, so more representative. In a self-generated gradient, you can see the cells, let's just show that again. The cells keep carrying the gradient with them. And as they keep carrying the gradient with them, so they keep getting a really steep gradient, and they can respond for ages. We've measured them going over centimeters. We think we could probably make them go over as far as 10 centimeters, which is enormously more than you could ever get with a fixed gradient. So what that means is if you ever see a long gradient anywhere in biology, the cells must be taking part of it. Now, why am I telling you all of this? Because if you look at the melanoblasts, the precursors of melanocytes, and thus the precursors of melanoma during development, they have to migrate over several millimeters. They emerge, in this case in a mouse, at embryonic day 13 or 12, they actually emerge thereabouts. They emerge from the neural crest, which is here, and they migrate through the dermis all the way around the mouse's body and then down the arms and legs. And if they can't do that, you end up with a mouse with a black back and a white tummy. And when you look at the cells doing that, you say it is obviously steered migration, but it is not simple steered migration. And if you go and look at what's happening at the leading edge, this is GFP labeled melanoblasts in vivo, an astonishing experiment by Olivia Susanto, a postdoc from the group. You see exactly as in my melanoma slide I showed you earlier, the front cells have direction and the rear cells have no direction. So our interpretation of this is that these melanoblasts are migrating to a self-generated gradient that is here. There's attractant here. There's no attractant here because the melanoblasts have consumed it. And the gradient is just over this local area here of about 300, 400 microns. So we come back to this question, why does melanoma metastasize? And the answer is melanoblasts, the precursors, the embryonic precursors of melanoma, are programmed to spread. They're programmed to spread from where they are to where they aren't. And that's what melanoma does when it's spread. And these cells are also must be using self-generated gradients. So they have all the machinery for making and responding to a self-generated gradient. The signal is different, we presume, between embryos and the later cells, but the mechanism, the self-generated gradients and the G protein coupled receptors and so forth looks identical. I just want to end by showing you some mazes. The mazes are so cool that I think you would like them, and they express very neatly what is going on here. So if you again go to a PDE-based model, and this time you make a maze. You make a complicated system of paths. Again, we've filled it with red attractant, and again, we've put all the cells in one place. And you see this extraordinary thing that the cells make their own gradient. And as they make their own gradient, they can actually explore their environment. The gradient forms by diffusion. And as the gradient forms by diffusion, the cells can tell the difference between an empty branch and a full branch, and they can interpret it. And if I was speaking to a biology department, I would have made you put up all your hands and say, how many of you would believe a model if I showed it? And something like half, maybe a bit less, would have believed it. But of course, it would be a trick question because we've done it. And here are real cells in a real maze doing exactly the same as the model. And it's not surprising because we know all the parameters. And in fact, you could only really build this maze if you had a model. Because there's an awful lot of different ways of building a maze, and to see something this simple, you need to know the parameters. So cells really can explore through a maze. And here's some pancreatic cancer cells doing exactly the same. They're not as good at it. It's not surprising. They're pancreatic cancer cells. But again, these cells that move right the way through here would not ever do that randomly. And again, we started with no information. There was no gradient here. The only thing in the starting condition is that the cancer cells come from the left, as if they were in a tumor. They have made the chemotactic gradient that is steering them through this complicated maze. Now, this is gigantically biologically relevant. I mean, the mazes are cool and they're artificial and they're nuts. But if you imagine an immune cell trying to find a tumor, it's there in your blood vessels. It gets signals saying that maybe there is a tumor and it should respond to it, but it has to move through endothelia. It has to move through tissues, perhaps, to find the tumor. And the problem is exactly the same. If you are the germ cells in a mouse, the germ cells are made in a different part of the mouse embryo or a fly embryo or a human embryo, or what have you, than the gonad. And during the course of embryogenesis, the germ cells must migrate through the embryo to find the germ cells. If you don't do that, no babies. And when your neurites connect to their synapses, they have to find one another through long and complex distances, and so on and so forth. And of course, metastasis. When cells metastasize from a melanoma into the bloodstream and out of the bloodstream across the blood-brain barrier into your brain, they are doing something that is exactly like those mazes, which is why they are so important. Now, I had one or two other nice maze slides, but I'm aware that time is short and I must be well behaved. So I think actually what I'm going to do is saw off there. And if anyone wants to see some more maze pictures and understand the complexity of mazes, they can ask me later. Thank you all very much.